Hello, I'm Dr. Maurie Markman from City of Hope, and I'm delighted to briefly discuss with you a fascinating paper that just appeared in the Journal of Clinical Oncology, and one there's actually been a lot of discussion about since the original abstract came out several months before. The paper is entitled Efficacy and Safety of Trastuzumab Deruxtecan in Patients with HER2-Expressing Solid Tumors. Primary results from the Destiny Pan-Tumor-02 phase 2 trial. This is a fascinating study. Antibody-drug conjugates, of course, have had a lot of interest, a lot of work that's been going on now for a number of years. And the concept of focusing on a particular molecular target and then potentially looking at the value of that drug across multiple tumor types, assuming the target is present, has really been, it's been a palpable discussion and within the oncology community. And this particular research study addresses this. So this drug that I noted has actually already been FDA approved in HER2 expressing breast and gastric cancers, as well as HER2 mutant non-small cell lung cancer. So the specific study here was looking at other tumor types, not the ones that are already approved, and in tumors that had immunohistochemical staining by IHC 3+ or 2+ by local labs were eligible for the trial. A number of tumor types were looked at in this trial, including endometrial cancer, ovarian cancer, cervical cancer, bladder, biliary tract, pancreatic, and others. I'm going to focus in a moment on the gynecologic cancers, which is my own clinical and research interest. The bottom line, the objective response rate in this trial overall in this population, patients who had at least one prior therapy before on the trial, or settings where there was no standard of care option. The objective response rate overall was 37.5%. And in those individuals who were 3+, the objective response rate was considerably higher than that. And as I noted, if you look at the data specifically in endometrial cancer, cervical cancer, and ovarian cancer, again, +2 or +3 staining, the endometrial cancer objective response rate was 57.5, cervical cancer 37.5, and ovarian cancer 42.5. That's percent. And when you then look specifically at the +3 staining, the actual objective response rates, remembering the numbers get small, was even considerably higher. I should also note that even though the eligibility for this trial was at least one prior regimen in the case of endometrial, cervical, and ovarian cancer, the patients on the trial actually had received, depending on the tumor type, at least 2 and sometimes up 3, 4+ regimens. So this is fascinating. So here we have a situation where evidence of overexpression of HER2 in the 3 gynecologic malignancies, we're actually talking to 3 different tumors types. We have objective response rates of over 40%, and those responses actually lasted a very reasonable period of time. Very soon, I think we can say that we will have a new therapeutic option for these tumor types in patients who have failed initial or standard therapies to date. And very importantly, we're talking here now about increasingly molecular-based therapies, small percentage of patients in each of the tumor types, but where that target is present, a very reasonable therapeutic option available for these patients. I'm going to encourage all oncologists, patients with these malignancies, their families, to see this paper, highlights of this paper, and wait for more information that's going to be coming out about this drug in the near future, as well as antibody-drug conjugates in general and molecular targeting therapy in precision medicine and oncology, which, of course, is the critical future of drug development in this area. And I thank you for your attention.